The cell membrane. To start off with the cell membrane, we start with the phospholipid head. The phospholipid is a molecule that constitutes the inner bilayer of biological membranes. So to start off, let's look at the big photo right here. This is a phospholipid head, the red circle. In my diagram, it's all these red dots. They act as the heads. The phospholipid head is polar and hydrophilic. Polar means that each end of it has a different charge that attracts and repels different materials. The phospholipid tail. It's the blue part right here, that is attached to the red circles. So it's attached to the phospholipid head. Again, a phospholipid is a molecule that constitutes the inner bilayer of biological membranes. So the bilayer is this whole section where they meet. The phospholipid tail is nonpolar and hydrophobic, so it does not like water. It repels water, while the head is hydrophilic and it attracts water. So the bilayer is this part right here. The tail right here are these blue lines, and the heads are the red dots. Carbohydrate chain. It's a small chain founding in the cell membrane. It assists in cell recognition, it stores energy, and it's a supporting structure. So here I circled the carbohydrate chain. On here, it's right on top. It's like the black cross. Glycoproteins. A glycoprotein is a molecule that consists of a carbohydrate plus a protein. So in the previous slide, I mentioned a carbohydrate chain, and that carbohydrate chain right here is connected to the glycoprotein. An example of when this is useful is in the immune system. The peripheral protein. It's a protein that is not tightly bonded to the cell membrane, and they can just sit on the surface, but they're anchored by a few of the hydrogen atoms. So it's in this diagram, it's the part of the protein that sticks out. So it's this part. It sticks out right here, sticks out above the phospholipid heads. The channel protein. This is inside the bilayer. Well, first of all, what is a protein? It's a group of organic compounds made up of 20 different amino acid chains, and the sequence of those chains determines the specific shape and function of the protein. So this picture right here, I enlarged a photo of what a channel protein looks like because it allows molecules to move through it. A channel protein is a protein that allows a specific substance to move across a cell. Integral protein, as you can tell by the term itself, integral, it's a protein that is inside the bilayer. Here I circled it, right there. Integral proteins are permanently connected to the cell membrane. Hydrophilic region. In my diagram, I drew it. It's from about this part of the bilayer, so the top part of the phospholipid head and tail, the top part right here, all the way up, they are attracted to water. So the region of the cell membrane that is attracted to water, the molecules are typically polar, which enables them to absorb water. So I enlarged another photo right here, and I circled the hydrophilic region. So as you can imagine, there has to be a hydrophobic region. The hydrophobic region is the inside of the tails to the outside ends of the tails where the heads meet. And it's the region of the cell membrane that repels water. So the inside does not want water. These hydrophobic tails do not want water, while the heads want water. They attract it. So how do hydrophilic and hydrophilic properties of phospholipids support the cell membrane? Well, let's review some of the properties. Hydrophilic means attracted to water, and hydrophobic means not attracted to water, but attracted to hydrophobic tails. So like I said before, in the bilayer, the tails are attracted to one another because they are all hydrophobic. So the cell membrane is made up of phospholipid molecules. They have a double layer. They have a double layer of phospholipid molecules. The heads face the water, the tails face the inside. The bilayer allows stability and flexibility. Phospholipids in a fluid state. So those are just like the basic key points I want to highlight, but now I'm actually going to explain what I am actually talking about. So phospholipid molecules, they make up the cell membrane, and they can either be hydrophilic or hydrophobic. They have a hydrophilic phosphate head and two hydrophobic tails. The cell membrane is made up of a double layer of these phospholipid molecules. So the tails face each other, and the heads are on the outside of the tails. So the cell membranes are made of a double layer of these phospholipid molecules. This is because in water, the hydrophilic heads will face the water and the hydrophobic tails will be in the center because they face away from the water. And that's given from the definitions themselves. The phospholipid bilayer makes the membrane very stable, but also allows flexibility. And because it's in a fluid state, it allows the cell to change shape easily. Now to explain diffusion. Diffusion is the passive movement of particles from a region of high concentration to a region of low concentration. So the purple dots right here demonstrate a substance that is outside of the cell membrane. The cell membrane is right here. It consists of all the heads and tails, the phospholipid heads and tails, and diffusion requires no energy, so it's a natural occurrence. The high concentration of purple dots, so where there is more purple dots, will move down through the cell membrane and go to a lower concentration. So it's almost like you're in a crowded dance room, and instead of bumping elbows, you want room. So you go from the crowded area, you walk through the crowd, and you go to a less crowded area. And that is basically diffusion. Okay, osmosis. Osmosis is the passive movement of water molecules across a partially permeable membrane from a region of lower solute concentration to a region of higher solute concentration. So in this diagram, I represented the water molecules with blue dots and the sugar concentration molecules with green dots. So on the left-hand side, there is a low sugar concentration, there's no green dots, and there's a high water concentration, there's a lot of blue dots. On the right-hand side, I demonstrated a high sugar concentration with a lot of green dots and a low water concentration with a few amount of blue dots. So in osmosis, this permeable membrane, the red line right here, the low sugar concentration wants to move where they have more room. So if you have a high and a low concentration of water, the substance, the sugar in this case, will want to move from the high concentration to the low concentration. It wants to even itself out. So the goal is for it to be in equilibrium. So the sugar concentration will move from left to right, from a high concentration to a low concentration, and until it equals out in an equilibrium. So facilitated diffusion. I demonstrated what facilitated diffusion would look like. 1 is where we start, and then we're going to go to 2, which I made a big diagram for, and then we go to 3. Okay, so facilitated diffusion happens outside of a cell, right here, and inside of a cell. And it is using a channel protein right here, which we used in an earlier slide. So the first step is almost like a hungry Pac-Man. He opens up and he takes some of the molecules that are outside of the cell and he takes it inwards. Right here, I demonstrated part of step 1. So as it opens, it channels the proteins, it channels the molecules through the protein and it spits it out like Pac-Man was almost too full. So facilitated diffusion requires a channel protein. This is just a quick overview of protein pumps and ATP in active transport membranes. So, first of all, what is active transport? Active transport involves the movement of substances through the membrane using energy from ATP. The advantage of active transport is that substances can be moved against the concentration gradient, meaning from a region of low concentration to a region of high concentration, where before it went from a high concentration to a low concentration. So since it's going from low to high, it's going to require an energy, an extra force to get the molecules moving across that membrane. This is only possible because of the protein pumps embedded in which it is used in active transport to move substances across using ATP. Each protein pump only transports certain substances, so the cell controls what comes in and what goes out. So each pump is special, so it has its own special purpose in this active transport. Okay, so we're going to talk about the sodium-potassium pump. It goes left to right to down to left to up. So we're going to start here, work our way across to 2, 3, 4, 5, and 6. So starting with 1, there is a high concentration of sodium outside of the cell membrane. And it's going to try to get a bond to the sodium-potassium pump so that it can get inside. So in the second part, the sodium binding stimulates a phospholipid action by the ATP. So the sodium is going to enter the potassium pump and it's going to start to leave. So the third part, potassium is released and sodium sites, they begin to be receptive again. So they're starting to take back in what was there before. And the fourth one, we are starting to see the protein to change in its conformation. So it starts to kick out the sodium. And then in the fifth part, the loss of the phosphate restores the protein's original conformation. So we're going from different concentrations and different shapes of the pump. The pump is the purple part, just in case you didn't know. And Na is sodium and K is potassium. And for the last part, the extracellular potassium binds to the protein, triggering release of the phosphate group. So in a more clarified way, in a shorter version, I'm going to explain how the sodium-potassium pump works. So the active transport involves the movement of substances through the membrane using energy from ATP. The advantage of active transport is that the substance can be moved against the concentration gradient, meaning from region of low concentration to region of high concentration. So in this scenario, sodium has a high concentration on the outside and potassium has a low concentration on the outside. So it's possible to move against the concentration gradient because the cell membrane has protein pumps embedded in which active transport moves the substances by using ATP. So this protein pump right here uses ATP, a type of energy, to move molecules from the outside of the cell to the inside of the cell, from the inside of the cell to the outside of the cell. So each protein pump only transports certain substances so the cell can control what comes in and out. And in this case, it's sodium-potassium.